Good morning, good afternoon, good evening to every one of us. So, the introduction for our presentation is physical evidence must be collected and preserved properly to remain admissible in court, meaning we need to ensure the proper collection and preservation until the chain of custody down to the court in order for it to be admissible in court and not to be dismissed as an evidence because evidence can make or break a case. Wherein in the presentation we will focus on the three important types of trace evidence or evidences that will link the perpetrator to the crime scene such as the soil, the plant materials, and the glass, where each can link the suspect, the victim, and the crime scene to forensic analysis. Why proper collection matters? Number one, because it prevents contamination or mixing of samples. Because once the sample is contaminated, it is already inadmissible because the lab results will show different from what should be shown, and maintains the chain of custody or the continuity of possession, which refers to the document and the unbroken transfer of the evidence from the moment of the collection down to the court presentation, and ensure accurate laboratory results. Because if we can prevent the contamination of evidences, we can ensure accurate laboratory results. And lastly, it helps reconstruct events and link the individuals to the crime scene. Okay, so soil evidence from the crime scene. The procedure here is follows. Collect about 10 soil samples, 2 from the question site and 8 from the surrounding areas, wherein you need to have 3 to 4 meter radius apart. And take samples from different depths and photograph the site, of course. And each sample must have zero point 0.5 deciliters, but the thing to be followed here is the collection of 10 cell samples. That is like the minimum by minimum because majority of the time it depends on the case that happened for the SOCO team. And then for the two on the question side and the surrounding areas, just imagine an inner and outer circle. And then for the outer circle, in order for you to have the 3 to 4 meter radius, it's better to just follow the north, the east, west, and south. And then on the taking of samples from various depths, I have asked some of my titos and titas working in the forensic department um, that they majority just collect organic to subsoil, but it really depends because if the liquid sample seeps down to the parent rock, they need to really collect down to the parent rock. But majority of the time, subsoil is just enough because majority of the time they said that liquid samples only seep into this alluviation layer and just take the subsoil for safety measures. As for the packaging and storage of the soil evidence, use a plastic or glass containers with tight fitting caps. But the best thing to use here is airtight seal containers because sometimes moisture can damage the liquid samples within the soil samples that is needed for the forensic analysis. And dry the samples unless sent immediately to the PNP crime laboratory. The best practice here, as was stated, is just to send it immediately. Don't air dry it, just send it immediately to the laboratory for forensic analysis or laboratory assessment. Now for the soil evidences under footwear prints. The procedure here is preserve the print and keep any soil from sticking off it and collect 8 or more soil samples from the nearby areas. If you have done forensic 3, you would have conducted the experiment regarding the plaster of Paris, wherein this is a chemical that dries up not quickly, but if it is properly heated, it can form the shape of the footprint wherein you can collect the type of print which is used in the forensic analysis. But do not forget to collect 8 or more soil samples from the nearby sites, from the nearby areas. As for the packaging, use to seal plastic or glass containers and labeled clearly, wherein you need to input the location, the depth, and the time collected. But actually, regarding on this collection, it is clearly questionable because it did not state how can you properly collect it? Do you need to use the plaster of Paris method or just take up the whole gram itself? But the method that the PNP is still using until today, wherein I ask, is because they are in modern province, so they still use the plaster of Paris to be exact, wherein you just need to collect prints from the area wherein you, not all, but like mini you need to still collect two of the prints, just the left and right foot, but at the different areas in the different times. That's all. Next, we have the plant material from the crime scene. The procedure is as follows: collect the whole plant if possible, including the parts that might stick to clothing, and photograph the site before collection. The best practice here is first photograph the whole site and just collect the whole plant. It's not possible just collect the whole plant. Just put it in in the vehicle and send it immediately to the crime laboratory. That is the best practice, because in the procedure it would state that you need to package it properly, but you just need to ensure that the plant that does not move sideways or tumble down or roll when it is being sent to the crime laboratory. And as for the packaging, it will be on the nice slide. This is the packaging and storage according to the PNP manual, wherein you need to wrap it in a fold newspaper and place it inside the paper sack. But the best practice here is still number two, wherein you need to transport it immediately to the PNP crime laboratory. Immediately, because in the on the transport of evidences, of course, the cover is number one, but this is number two, especially if it has liquid samples, biological samples, so on and so forth. That's very crucial on the case, because if it is leaded airbag and placed before a firebag, why do we need to place it in a firebag? Because a firebag is a controlled environment. In order for the no degradation that will happen if we just like put it in an airbag, it will just moisture up and maybe damage the plant and the sample itself. For glass evidence from crime scene, for reference purposes, just remove a 1 centimeter squared piece of glass from each window pane and collect all the broken glasses for physical matching. Use a tweezer or forceps with the plastic tip, and for the packaging, it's better to fold it first with the newspaper or any paper packaging before putting it in a cardboard box to prevent it from breaking any further. Now for glass, in order to determine the breaking direction, if possible, remove the whole sash, secure loose pieces with tape, and collect all fragments. The loose pieces with tape, do not use tape in order for collection, but for just like the area wherein it is been broken, or kung saan nabasag ang glass, just put a tape on it to prevent further breaking and mark its side to show which side, if it is inner or outer, or where it came from. Well, now for the packaging, it's still the same with the glass evidence from the crime scene, wherein you use paper foldings or paper wrappings before putting it in the cardboard box to keep the glass from breaking any further. Now for glass from clothes and footwear, the procedure states that pick up the pieces with the forceps or by vacuum cleaning, but the vacuum cleaning, it's better to use the handled one rather than the big vacuum, and send the shoes or clothes immediately with the glass fragments to the PNP Crime Laboratory as fast as possible. For the packaging, we have three types here. Two types. Small fragments should be folded in the paper and plastic tubes or boxes. Now, for the folding, you can use the apothecary method, wherein you can just search it because I cannot show it here, sorry. And for the large pieces, just pack it in a plastic box. But yeah, just pack it in a plastic box. Never use tape to collect fragments. That's all. Now we are at the end of the reporting, wherein this is just a conclusion. Now we need to ensure the proper handling of the evidences as was stated in order to have the integrity of the evidence, because all of the time integrity and chain of custody is being questioned in court in order for the admissibility of evidence, which is number three. Because if you don't have the integrity of evidence, then it's biased, it's contaminated, then your evidence is not admissible, even if it is like the primary evidence. that can be used against the case. It will just be inadmissible, and all of your hard work and the hard work of your team, the SOCA team, and then the crime laboratory will be just useless and unreliable forensic analysis. You need to ensure proper handling and prevent contamination, because you cannot have a reliable forensic analysis of forensic result or laboratory result if it is damaged or contaminated in the first place. Small quote here is: every small trace can serve as a silent witness in criminal investigation or criminal procedure, wherein just that dollop of blood that you have spotted can link the suspect to the crime scene. It helps and became that small blood became the number one priority that connected the perpetrator to the crime scene. Now, thank you for listening. Good night, good morning, good evening, and good afternoon. Thank you for listening.